Hello students, in this session we are going to study about radioimmunoassay. It is one of the analytical techniques used for quantitative analysis of the biomolecules like steroids, hormones, peptides, etc. So now let us see what is meant by radioimmunoassay. So as you can see, it is made up by combining three terms, that is radio, immuno and assay. So by radio we means use of radioactive material. The materials which emit radiations like alpha, beta and gamma. Second term immuno. So immuno generally stands for antigen antibody binding. And assay, it means detection of compound, or you can say to find out the percentage purity. So a radioimmunoassay is an immunoassay that uses radio-labeled molecules in a stepwise formation of immune complexes. It is a very sensitive in vitro assay technique used to measure concentrations of substances like hormone levels in the blood by use of antibodies. Let us see how this technique originated. So radioimmunoassay, it was developed by Rosalind Yellow and Solomon Burson in 1959. So when they invented this technique, at that time the purpose was to measure the level of insulin in the plasma. It turned out that the level of hormone in the blood could be checked by an in vitro assay. In 1977, they also received the Nobel Prize for the development of RIA, that is radioimmunoassay. So what is the importance of radioimmunoassay? Now it is very difficult by the ordinary analytical technique to measure the concentration of biomolecules. Thus RIA has revolutionized estimation of several compounds in biological fluids that are found in exceedingly low concentrations. So biomolecules, they are found in very low concentration, generally in nanogram and picogram level. So their estimation has been made possible by use of radioimmunoassay. It is a highly specific and sensitive analytical tool. We know that antigen antibody binding is also very specific. Thus, this technique is specific and sensitive. Then compounds like peptides, steroid hormones, vitamins, drugs, antibiotics, then your nucleic acids, structural proteins and hormone receptor proteins can be analyzed by using radioimmunoassay. So before saying the principle, let us see some of the important terms which we are going to use. First is antigen. So we all know that antigen is the substance that the body is trying to fight off by mounting an immune response. Okay, so the foreign substance which a body tries to fight off, and in doing so the body generates some immune responses against that foreign body. So that foreign body is termed as antigen. Next term is antibody. It is a protein that is produced by the body in response to an invading substance, that is to fight off the antigen, or you can say foreign substance. Thus, antibodies are produced as a part of body's immune response to protect itself. Next term is immunoassay. So you can again here see it is made by immuno and assay. So it is a biochemical test that measures the presence or concentration of a macromolecule or small molecule in a solution through use of an antibody or sometimes antigen. Analytes in the biological liquids such as serum or urine are frequently measured using immunoassays for medical and research purposes. Next term, radio-labeled molecules. Okay, so as you know, radioimmunoassay, we are going to use here the radio-labeled molecules. So what are those? These are chemical compounds in which one or more atoms have been replaced by a radionuclide. That is, you can say the atoms or molecules which emit radiations like alpha, beta and gamma. Next is analyte. So analyte is anything measured by a laboratory test. So when we are talking about immunoassay, so in immunoassay testing, the analyte may either be an antibody or an antigen. So we can measure either an antibody or an antigen. Immunoassays utilize one or more selected antibodies to detect analytes of interest. Okay, now what are the analytes which we can measure through RIA? So they are naturally present in the body, such as thyroid hormone. The analyte could also be the substance which is not typically present in our body in normal conditions, such as cancer antigen. So they will be produced only when the cancer has invaded the body. The analyte, it may also be the substance which does not occur naturally in the body, such as abused drug. Now let us come to the principle of RIA. So now we know all the terminologies like immunoassay, antigen, antibody, what are radiolabeled molecules. So it is easy to understand the principle. So you can here see the terms antibody, then labeled, unlabeled antigen, competitive binding and measurement of radioactivity. So RIA combines the principle of radioactivity of isotopes and immunological reactions of antigen and antibody. Its principle is primarily based on the competition between the labeled and unlabeled antigen to bind with the antibody to form antigen-antibody complexes. Okay, so here there occurs a competitive binding between labeled and unlabeled antigen to bind with the antibody. Now you know that antigen and antibody, they combine to form a complex. Okay. Now if we use two types of antigens like labeled antigen and unlabeled antigen, they both are going to compete with the antibody for binding, because the both are same, labeled and unlabeled, they are same antigen, only the difference is one is radioactive and other is non-radioactive. So the competition occurs between them for binding with antibody. Okay, so let us see here, unlabeled antigen is the substance, say for example insulin, to be determined. Antibody is produced by injecting the antigen to goat or rabbit. Now suppose we want to determine insulin, okay, from a patient's serum or from his blood sample. So what we will do, we will take a unlabeled antigen, that is the standard antigen insulin, known concentration of insulin we will take, and we are going to inject it into the goat or rabbit, so antibodies will be produced. Now these antibodies we will utilize for our RIA procedure. So specific antibody is then subjected to react with unlabeled antigen in the presence of excess amount of isotopically labeled antigen with known radioactivity. Okay, so now we are having antibodies. We will take a standard antigen whose concentration we already know and which is unlabeled, and then we will take a labeled antigen, that is antigen which is made radioactive by use of a radionuclide. Okay, so suppose for example this is your insulin, unlabeled insulin, let us say, and this is your labeled insulin, and these are your antibodies. These antibodies have been produced by injecting insulin into some animal. Okay, so these are your antibodies which are specific to the insulin. So now what will happen? There occurs a competition between the antigens, that is unlabeled as well as labeled, to bind with antibody. Now here you can see there is unlabeled insulin, labeled insulin. So there will occur competition between the two for binding with the antibody. The labeled antigen will have upper hand due to excess presence. Now initially labeled antigen is in higher concentration. So what will happen? Labeled antigen will bind in higher concentration as compared to the unlabeled antigen. Okay, this is going to bind in the higher concentration. But what happens as we slowly increase the concentration of your unlabeled antigen, what will happen? The labeled antigen will start getting replaced with unlabeled antigen. Okay, so as the concentration of unlabeled antigen increases, the amount of labeled antigen antibody complexes decreases. Thus, the concentration of labeled antigen and antibodies inversely related to the concentration of unlabeled antigen. So, as the concentration of unlabeled antigen increases, the concentration of labeled antigen antibody complex start decreasing. You can see it is the inverse relationship. And this relation is almost linear. Thus, radioimmunoassay determines the concentration of antigen in the sample on the basis of competitive binding between the unlabeled antigen and radiolabeled antigen for specific high affinity antibody. Now see, when we see this diagram, it will be more clear. The principle will be more clear. Now see here, these are the microtiter wells which are coated with antibodies. Okay. So initially we will take the radio-labeled antigen. You can see these are the radio-labeled antigens. You can see the binding between antibody and antigens. Then washing is given. Why washing is given? So that the unbound antigens they get washed away, so that they will not cause interference in the results. Okay. Now if we add the unlabeled antigen, you can see there will occur the competitive binding between both of this. And here you can see some of your labeled antigens have been replaced by the unlabeled antigen. So what is going to happen? Radioactivity decreases. So initially there will be 100% radioactivity. Okay, but now the unlabeled antigen have replaced the labeled antigen. So here you can see there is the decreased radioactivity. Radioactivity gets decreased. Okay, and by using this principle that how much radioactivity has got decreased, we can find out concentration of unknown antigen, that is antigen from your sample. So what we are going to do? We are going to plot the curve of radioactivity versus concentration of antigen. So here you can see as the concentration of antigen increases, that is here we are talking about the unlabeled antigen. So as the concentration of unlabeled antigen increases, the radioactivity decreases. Then what we can do? We can take our sample antigen, and by measuring the radioactivity, we can extrapolate it on the x-axis and we can find out the concentration of unknown antigen. Okay, so a standard curve can be drawn by using different concentrations of unlabeled antigen and same quantity of antibody and labeled antigen. The labeled antigen antibody complex is separated by precipitation. and the radioactivity of the antigen antibody complex can be determined. Let us see the procedure. So by procedure, the principle will be more clear that what actually we are going to do in the radioimmunoassay technique. How we can find out the concentration of unknown sample. So take microtiter wells, coat them with the fixed amount of antibodies specific to the antigen to be determined. Now add a fixed amount of radiolabeled antigen to the first well. And this well is known as the control well. Okay. Rinse the well to remove the unbound antigens. Measure the radioactivity of the first well. It will be maximum, say 100%. Okay, so see what we have done. We are going to take microtiter wells, coat each of the well with the specific fixed amount of antibodies. Okay, concentration of antibodies should be same in each of these wells. Now in the first well, what you are going to add? Add radio labeled antigen, and its quantity should be such that all the binding sites on the antibodies get saturated, and this well will be known as control well because this well contains all the labeled antigens bound to the antibodies, and its radioactivity will be considered as 100%. Now to other remaining well, add same amount of radiolabeled antigen. Again you are going to add same amount of radiolabeled antigens. Okay. But along with this radiolabeled antigen, what you are going to add? You are going to add unlabeled antigen in the increasing amount. Okay, so along with your radiolabeled antigen, add unlabeled antigen in the increasing amount. So this well number 3, it should contain higher concentration of your unlabeled standard antigen as compared to the well number 2. So now what will happen? There will occur competitive binding. So wait for half an hour. Antibodies cannot distinguish between the labeled and unlabeled antigen. So during this half an hour, what will occur? There will occur competition between the labeled and unlabeled antigen for antigen binding sites on the antibodies. Now in this well too, some of the labeled antigen will be replaced by unlabeled antigen. So you can see that radioactivity will decrease. Again in well 3, radioactivity will decrease more because it contains higher amount of unlabeled antigen as compared to well number 2. So unlabeled antigens will occupy more sites on the antibodies with increasing concentration. Each well will now contain bound as well as unbound antigens. So we will have to wash it to remove the unbound antigen. Now measure the radioactivity of each well with gamma counter. Okay, so gamma counter, it is the instrument used for measuring the radioactivity. Radioactivity will decrease in each well with increasing concentration of unlabeled antigen. There will be less amount of labeled antigen bound to the antibodies as compared to the control well. At the end, we have radioactivity of each well. Also, we know the concentration of unlabeled antigen in each well. So, on the basis of above information, plot the standard curve. So, the curve we have to plot between the concentration of antigen, that is unlabeled standard antigen, and radioactivity. As the concentration of unlabeled antigen increases, radioactivity, you can see, decreases. Okay, so now we have got the standard curve. Now what we are going to do, take a microtiter well with same amount of antibodies as we have taken to prepare the standard curve. Now add to it fixed amount of labeled antigen as that taken for the standard curve. Along with this, now add the suspension of patient's serum, that is antigens to be determined. These antigens will compete with labeled antigens for antigen binding sites on antibodies. Again, rinse the well to remove unbound antigen and measure the radioactivity. Extrapolate this value of radioactivity on the standard curve. In this, we will get to know the concentration of antigens in patient's serum. Now you can see this was our standard curve. Now by extrapolating this radioactivity on the x-axis, now which is this? This is the radioactivity of the well containing patient's serum. So on extrapolating on the x-axis, we will get to know the concentration of antigen, that is antigen present in patient's serum. So this was about the principle and procedure of radioimmunoassay. Thank you.